Welcome back to Quantum Conversations. My name is Sarah, and today my co-host Carrie Bennett and I have a very special two-part episode with Glen Jeffrey, who is one of the world's leading experts, scientists, and researchers on how light interacts with the mitochondria, our metabolism, our body's ability to heal and repair, and so much more. Now, Glen has really been studying how, in particular, red and infrared light impact the mitochondria. And again, things like our metabolism, blood sugar responses, insulin. He also has studied blue light extensively, as you'll hear in this interview, and how that can actually cause people to gain weight and have blood sugar issues. So these are topics that Carrie and I discuss all the time here on Quantum Conversations. And Glen is just a really lovely, fun person that we thoroughly enjoyed speaking with. Again, this is going to be divided into two episodes. So make sure you're subscribed so you can catch both parts. And quickly, I wanted to let you know, Carrie and I are extending our VIP Health Transformation Summit upgrade deal. Now, this deal allows you to get about $2,000 worth of courses for $99. I'll put a link in the show notes and in the information section for you so you can check out some of those. But through this link, you'll be able to get my 3-day leptin kickstart, my circadian health for the busy person, Carrie's nervous system and nature course, and again, about 12 other courses from creators who were a part of our Health Transformation Summit. And you'll also get all of the recordings in case you missed them. So again, that'll be in the pinned comment as well as in the show notes. I hope you enjoy this episode with Glen Jeffrey, and I will talk with you again soon. Hello, everyone. Welcome back to Quantum Conversations. We are very excited to have a very special guest today here with us that is really going to expand on the light story. Carrie, do you want to do a little bit of an intro and then we'll let Glen just start chatting away?
Sure. So we have the lovely Dr. Glen Jeffrey, and I've came across his work originally when he was on Max Gullhane's podcast, a regenerative health podcast, which is a lovely podcast and was an excellent interview. And I said to Max, I was like, I gotta get to know this guy because I'm about to present at the water conference, in Lisbon, and Jerry Pollack's going to be there. And I'm speaking specifically on mitochondria. And essentially mitochondrial water and ATP production in response to different lighting environments. And so that I was so excited to then have Glen reach back after that introduction and then give me a whole bunch of research to dive into, which was so much fun to then to be able to present in Lisbon. And so this is right up the alley of all of our people, Glen. Your work is it's cutting edge, yet I think that there's a lot of practical takeaways that we can come up with for our people. And so we're so excited to have you. So can you just give our listeners a little bit of background about who you are and how you came to study mitochondria and their interaction with light?
Oh, well, it's a tortuous story. I originally went to university slightly late and read experimental psychology, but that was at a time when experimental psychology was a lot of physiology and a lot of statistics and maths. I went on to do a doctorate in Oxford, and I really was in the area of vision and neuroscience. I spent far too much time in Oxford. But still profoundly in vision and in visual bits of the brain. I eventually got a permanent faculty position here at the Institute of Ophthalmology, University College London. And that brought me into contact with a different group of people, brought me into contact with the patient population because we're part of an extremely large Moorfields Eye Hospital. And so people started asking me a few questions about some very, very strange medical conditions. And then, as you get older, your interests shift a little bit. When I was a young graduate student, I was interested in development. Now I'm getting old, I'm interested in aging. And the one thing that bounces up every time if you're interested in aging is mitochondria. And I went to an extremely impressive seminar by a guy called Ilias Tachtanides, who's an engineer. And what he was doing was he was shining a light through the head of neonatal children that had stroke. And that light, he was looking at it coming out the other side, and he was making predictions about survival by looking at their mitochondria. And I said, hey, if you can do that going through the head, do you think you could do that in the eye? And he went, yeah, it's not a problem at all. Then the ball started rolling. I started getting to mitochondria. I learned how to spell the word mitochondria. And I ended up in a situation now where 90% of my lab's research is mitochondrial. It includes odd things, not just aging, children with mitochondrial disease, people with things like macular degeneration. Generally, if you're interested in aging, mitochondria grab you at every corner. They are core to it. And when I learned that mitochondria make ATP, and when I learned that you make your own body weight in ATP every day, you start picking up on things, thinking this is the place to be. So we've expanded. We're doing different things. We're spending lots of time working with architects now, working with lighting engineers. So it's, some people spend all their lives working on this particular receptor in the brain. I don't have a personality for that. I kind of bounce around a little bit. And so suddenly working with architects, it's really an interesting, different thing. They dress nicely. They've got really nice classic cars. It's a different culture altogether. So that's roughly who I am and where I am today.
It's so interesting. And yes, mitochondria, I was kind of hooked too when I realized how integral mitochondria were with all aspects of our physiology. And so it's exciting. And what I've really loved as you kind of started to lay out your research is that you indicated, and I think this is something that our listeners would be interested in, is that the light response, or how mitochondria respond to light, is conserved across species. We sometimes talk about, we kind of poo-poo sometimes where there's research that's been done on ultraviolet light using, let's say, an arc lamp and shined on like a hairless rat. And, it's you trying to make the translation between this ultraviolet light exposure to a nocturnal creature showing damage to ultraviolet light and how that can translate then to UV light being harmful to humans. I don't find that to be an apples to apples comparison. But what I find interesting is that the optics in mitochondria are apparently conserved across species, whether you're talking about a Drosophila or a bee or, a human being. So would that be a true statement?
It's definitely a true statement, and it's quite surprising. I mean, I'm quite surprised by it. Anything I find in a fly or a bee translates to a mouse. And then when we try to do that clinical human translation, it doesn't let me down. That story still seems to be the same. Now, there are little tweaks in it, I'm sure, but it's not just the big picture, it's the intermediate picture that has got this conservation of mechanisms all the way down. Now, that's great for me because if I want to do a population study, I can't do that with mice. I can't do that with humans. If I need 150 of this and 150 of that, first of all, mice in the UK cost about $5 or $6. They cost, so many dollars per day to keep. And I work a lot on aging. So I've got to keep them for a long time. So I've always been someone who's very comparative in the sorts of things they do. I have another side that people don't see too much, which is working on Arctic and Arctic light. And we spent ages working on reindeer and how they coped with darkness in winter. So I'm really happy to make that jump. And yeah, it is highly conserved. When you think it's such a fundamental building block for life, maybe it's not too surprising that it is as conserved as it is. But I've yet to find a significant thing where there's a difference between the fly and the human.
Oh, fabulous. So interesting. So, okay. So this is now where we get to dive into some fun stuff, I think, for our listeners in terms of what have you noticed in terms of the shift from, I guess, the more incandescent bulbs, which are dominant in the red and the infrared. What can you say about how mitochondria may be negatively impacted by these blue dominant bulbs that we have these days? What are some things that you're seeing?
Well, it's slightly disarming that the model is simple. The model is incredibly simple. So if we think about the mitochondria as a battery, that's how I always explain it. My mum gets that when I explain it to her, which is a good yardstick. the blue light discharges the battery. It reduces the charge. The red light increases the charge. Now, for millions of years, we've wandered around under sunlight where the balance between the blue and the red is pretty, it's been in balance. Sunlight hasn't changed for billions of years. So we're wandering around in that. And then really since the early 2000s, everything's changed, because the light that came out of an incandescent bulb was very similar to sunlight. And prior to that, we had candles. Suddenly, boom, there's a massive change in lighting. And the thing that I think is terribly surprising is our mitochondria see it. They're aware of it because they respond both to blue and to red light. Now, the blue light does tend not to go very deep in the body. But the red light, a few months ago, we were measuring people standing out in sunlight, and we were putting sensitive devices on their back, and we were measuring the sunlight coming through their body. Those long wavelengths go through. And if they go, we didn't measure much coming out, but that's important because it means that the light as it goes in isn't reflected. It's going in, it's staying in, it's doing something, and I'm measuring this little bit that comes out the back. So great if we're in sunlight. If you're sitting in your office, with standard lighting, that light that goes through your body, you're not getting it. And then you start looking at all the population data, how certain diseases change when you don't get much sunlight. So that's driving me now. And we've just finished a study, great study, done by one of the people in the lab. And he's gone to a big building in UCL here in London, and it's got no daylight. And we measured the retinal sensitivity of the people working in it. And then we gave them all incandescent lamps. And we left them for 2 weeks. And we came back, and the result was really dramatic. Their ability to detect the differences in colours had improved very, very significantly. So then we did the reverse experiment. We took them away. And slowly, their ability to detect colour differences slowly declined. Wow. So it was an experiment that worked both ways. Now, we concentrate on vision because it's my background. It makes my director happy because this is an institute of ophthalmology. He's not so happy when I work on bees and flies. But this is really quite important. People in that environment, and I believe many others, are working in an underpowered situation. Now, we want to go and do what we've done in other situations, which is want to test their blood sugars, because we know red light makes your mitochondria work. When your mitochondria work, they need food, and their food is glucose and oxygen. And we've shown that red light reduces your blood sugars. That has implications for diabetes. We're getting back in there, and we're looking at the blood glucose in these guys sitting in this environment as we chop and change the lights. And the great thing about that experiment is it doesn't cost very much. You just get the light bulbs and you change them. And then everyone gets very fed up with you when you take them away because they see what's happened to them. But it's a great, simple experiment.
So are the changes in visual color perception a direct reflection of the health of the mitochondria in the eye?
You've got more mitochondria in your eye than you've got in any other tissue. Your photoreceptors are just packed with it. And the reason for that is your visual sensitivity, your range, goes over 9 log units. From the darkest room you can see into the brightest sunlight. Now, we don't pay attention to that. We go from room to room with a 2 log unit change in light, and we just don't pay any attention. But your mitochondria have got to provide the energy for that adaptation. And so using the visual system is a pretty good model. It's got a great readout, which is, can you see it? Can you not see it? I can measure it. It gives me numbers. It makes biological sense because I just know all the mitochondria there. Little things are really helpful. Like you've got very few mitochondria in your blue channel, your ability to see blue. So we can watch the blue suffer before the red does, and then we can bring it back up and swap the two around. It's a nice system.
It's fun. And you can make probably some really pretty graphs in your publications showing these shifts and changes.
Colors. Yeah. Yeah.
Okay, so you alluded to blood glucose, which I think would be a fascinating thing to go into. Sarah is huge on metabolism, and our community loves talking about metabolism. And one of the things that everyone gets stuck on is, well, what should I be eating? What should I be eating?
Right.
What should I be eating? And we've been kind of, along with other people in this circadian space, have been saying, but what about the lighting? Because we do know that in these blue dominant light environments, you can have elevations in blood glucose in spite of not eating at all. And there's pathways in the brain that actually can lead to that. And so it was brilliant to come across the research that you've done showing that blue inhibits the mitochondria. Makes sense, meaning that the blood glucose has a tendency to stay potentially elevated because the mitochondria cannot use it efficiently.
Yeah.
Which has ramifications for diabetes. And then the red allows those mitochondria to use it all of a sudden and become efficient. So can you talk about some of the studies that you've done in, for example, glucose tolerance testing, and how red light might have been impactful in those instances?
Yeah, I mean, it came out of a car drive with a colleague when we were going to do a project somewhere else. And he turned around and said, Glenn, if we're doing what we think we're doing, then we should be regulating blood glucose by making those mitochondria work harder. And that involves human subjects. It involves doing glucose tolerance tests, which are really horrible, drinking loads of glucose, having your fingers pricked. And I said, well, I'm not sure about this. Let's do something else first. So we generally work on bees quite a lot. And so we took a whole group of bees and basically gave them a glucose tolerance test. We starved them overnight and we gave them a big chunk of glucose in the morning, which they ate like crazy. And they either had blue or they had red light. Now, the bees that had the red light, their blood glucose level, it's called hemolymph, their blood glucose level went down. The red light drove it down. The blue light took them straight into diabetes. So, it was frighteningly clear. And it was a super straightforward experiment. Remember, I'm pushing certainly the point that there's conservation of mitochondrial function. So having looked at that, we went, well, we can't give people blue light. We just can't give people blue light. So we did standard glucose tolerance tests on people. We looked at their vision, but we looked at their blood glucose level. So we got them in early in the morning, gave them red light or didn't give them red light, gave them a horrendous amount of glucose and pricked their finger every 15 minutes. Result was very clear. It is not necessarily so much of a problem if you've got high blood glucose. What is a problem is it's spiking. Right, you really want to get rid of those spikes. Spikes were way down, way down. One of the other things about these kind of experiments is you really get to know who your friends are because you need the subjects. Because we also stick a tube up your nose and measure your CO2. So after we did it on ourselves, the next one in line was my son, who I dragged in. And actually, that was a bit of an effort to get it done. Result was clear, absolutely clear. But these were ordinary, healthy people. We're now just finishing off the type 2 diabetes, and we've got exactly the same result. So red light can control blood sugars. It's not the only thing. You do have to pay attention to what you eat, and you do have to pay attention to being too sedentary. However, red light as part of the package to reduce diabetes is, I think, probably very important. And look at the big picture. If you look at populations that spend more time in sunlight compared to populations that are inside, the people inside have got lower ability to control their insulin regulation. So there's thousands of people there, and then there's a few of us sitting here in the lab sticking pins in our fingers and taking blood, but it all fits together. Although I would say, I keep saying to everybody who comes in, I can give you a bulb, I can do this, go out in the sunlight. Just go out in sunlight. Make sure you, particularly this time of year, when I come to work, it's pretty dark. When I go home, it's completely dark. To have that walk in the park at lunchtime. Yeah.
Mm-hmm.
The perfect recommendation. And what I want to make clear about this particular research study was that these participants were pre-treated with red light at 670 nanometers, I believe, to be correct. So how interesting, right? That it's not necessarily that you have to be in front of a red light panel the whole entire time. That it's this pre-treatment is essentially programming the mitochondria to be more metabolically efficient, so that when they do have that glucose exposure, they're able to metabolize it more efficiently.
Yeah. Now, I think there are a number of problems with this, and they came out from the vision problems, and that is that it's a surprising amount of red light, a surprisingly small amount of red light that is having this effect, you know, and that worries me. It worries me because the model isn't following a normal biological model. There's no dose response curve. It's a switch. Oomph, something happens. You give the red light, and then maybe an enzyme is produced somewhere. Now, that's fine. It works. It's a fantastic patch over a problem. But that's very different from daylight. When you're out in daylight, you're getting it all the time. So maybe the difference is I've got you in a blue environment, I'm giving you red to pump you up. Whereas when you're going out there, you're getting that natural balance between blue and red. But the take home has to be light is regulating blood sugar levels.
Yeah, absolutely. Now, I'm curious because in other studies too, this one I believe was in Drosophila, where you shined a small amount of blue light as well. It decreased mitochondrial membrane potential and potentially their motor capabilities as well. And it lasted for some duration. Was there a specific time of day that you kept consistent with that?
We do, but just because of the patterns of our lives. We tend to do experiments in the morning. You come in, you have a cup of coffee, you stand around wasting 10 or 15 minutes, and then you tend to go in the lab and do things. And I don't know why, but for a lot of scientists, they tend to do stuff in the morning, and the afternoons they're sitting in front of a computer. So we always did our experimental manipulations in the mornings, okay? Really did. And we're absolutely fine with that. We were very happy. But there were lots of people who were not getting the same results as us. And there was a big meeting, a vision meeting in Baltimore, maybe about 7 years ago. And we all knew one another. And one of the great things about this community initially was we shared data because there was so much low-hanging fruit. And we sat around and we talked. Why were you using males? Were you using females? And then it suddenly transpired that the big effects were always coming in the morning, right? When we worked that one out, everyone went away and a lot of people replicated one another's experiments and same effect. So whether you're a fly, a bee, or a human being, the red light experiments really only come to their optimum is if you do the exposures in the morning. The blue is a bit more messy. And I haven't really got my hand on that. And to some extent, I know where we're going with the blue. We've got to get rid of it. We've got to change the LEDs. I don't see why I've got to torture people and try and find out the time of day. There's so much stuff that we can do. Everyone that I'm talking to in my environment, the architects, the medics, the lighting engineers are all on board for getting rid of the blue. So we've won that one. Conceptually, we've won it.
Great. That's great. So I'm just, I don't know, Sarah, maybe this would make sense to you as well. I'm just wondering if, since at dawn and sunrise, we know that that's a fairly dominant in the red and infrared spectrum. I'm wondering if it's like the brain and the mitochondria, the skin is just the eyes are just used to having a dominant input of red, or perhaps even campfire before sunrise, but just a dominant input of red and infrared prior to the appearance of blue. And that's just something that triggers like a nonlinear effect.
Yeah. And nonlinear is a word I don't use very much, but you're absolutely right. You're absolutely right. It's a switch. It's nonlinear. There's no dose response curve. I always try and go back and think about where were we in evolution and why is this important in an evolutionary context? It's got to have something to do with sunlight. Mm-hmm. The morning is just so important because all your physiology is different in the morning. So I can really only drive up mitochondria in the morning when they're dynamic, when they're producing a lot of ATP. I can't do it late in the afternoon. In fact, I can't do it really after lunch very well. But then when we discuss it, when you wake up, during evolution, you're vulnerable. Someone could be watching you. you need to wake up and get stuff on the road. So your ATP peaks naturally anyway. Your hormones change like crazy and your blood sugars go up. A lot of diabetic people will tell you, they have a challenge with breakfast, because your blood sugars are harder to control. Now, I find that myself. I've got a complete map of my blood sugars now, and I know that I can watch my blood sugars go up if I have 2 croissants in the morning. But when I go home, by the time I've cycled home in the evening, I could eat piles of chocolate, and my blood sugars really don't change that much. So there is a lot in the time of day. There is a lot in circadian events. Mitochondria have got their own clocks, and I'm not always sure what they're doing, as well as there being a master clock and clocks in other tissues. But I'm not a circadian person.
Sure. No. It makes sense, so that if we kind of look to nature to see what is the information that we can, how can we interpret this data essentially through the lens of nature and evolution? I think that's typically what I always try to do.
Yeah. No, I'm if I can pin it down to something in terms of evolution, I will do. And a great physical anthropologist walked through my door, I don't know, 18 months ago, and Nate Domney, and from Dartmouth, and he came along and he said firelight, and firelight had been in the back of my mind. And he said, what was firelight doing? Well, the spectrum of firelight is very heavy in the red. I'm really unsure of where we stand on this one, but it is important, because when we evolved originally in a forested type environment, lots of trees. We then moved out into savanna, where we got a lot more sunlight, particularly on our heads. Big transitions change in human evolution with fire. I mean, the obvious ones are, your food's easier to digest. Your food doesn't go off quite as easily. Nate came in and said, there are other angles here. What are the other angles? So it's winter coming now. I bought half a ton of logs in summer, and I bought a fire pit. So, of course, when blokes go out and they light fires, they also have their cans of beer. They can't do it without it. So what we're going to do is we're going to sit there, and I'm going to get the people to sit around, and I'm going to be measuring if I could try and find out if I can measure firelight coming through the body by putting that instrumentation on people's backs.
Oh, I love that idea.
We want to talk a little bit about why we both use more red light therapy in the winter really quickly.
Yeah, absolutely. Come summer, it is so easy for me to pop outside for 5 minutes here, 10 minutes there, take the clothes off, and just soak up the natural light, right? And I know you do. I know you do the same. And it's beautiful because we're getting all of those red and near infrared wavelengths from the sun and imparting all those healing benefits from sunlight. But come, November, December in Michigan, there's very little opportunity for me to do that. It's just too cold, and so I'll go outside and get my light signaling through my eyes, but my skin really, that infrared and red that I want to absorb through my skin is just lacking. And so that's why I would highly recommend, if you live in an environment like that, you consider using a red light therapy panel in the winter. It can be the biggest game changer.
Yeah. And we both love the Boncharge Mini. This is super convenient. You can take it with you traveling. You guys don't know, when I'm recording the podcast, half the time I have it on my low back or somewhere in my body to mitigate some of this blue light. And for the months of November and December, you can use the code QuantumPod25 for November and December only to get 25% off the panel or anything on the Boncharge website. Carrie and I are both big fans.
Yeah. And one more thing really quickly with that. It's a great gift too, right? If you want to start getting people involved in this lifestyle and say it's the most convenient way, if there's a mom that who needs to tend to boo-boos and aches and pains for kids, or earaches, tummy aches, sore throats, it's a phenomenal gift to give as well, because it really can wipe away pain very fast.
I love it. All right, check it out. Link is in the show notes.
I think it's just, that's really interesting.
Sure.
Yeah.
Then our nighttime sun exposure, essentially, if it does come through in a similar way as the sunlight, I think in terms of the fire, I study extensively mitochondria and essentially water in the body. And mitochondria, we know, I'm certain you're aware of Scott Zimmerman's work and the near infrared component of stimulating mitochondrial melatonin, which is by far the biggest source of melatonin in the body. But also, are you aware of Dr. Gerald Pollack's work on exclusion zone water?
So when someone first asked me that question, I thought, hang on, who is that person? And I had to say, hang on, I think I know the name. And he very kindly contacted me, and I became much more comprehensively aware of what he was doing. And I have to say, I'm slightly ashamed that I didn't know him as well as I should have known him, but I'm getting there.
Yeah, it's just another layer, right, of what I had to unlearn in undergraduate and graduate school about how water behaved in the body, where it was kind of discarded as just being there. And now we know that it preferentially absorbs, at least exclusion zone water, preferentially absorbs light at about 3,000 nanometers. And that actually can act, again, as a reservoir of energy and charge in the body because it's negatively charged. So I'm just wondering if that's how we got that energetic source at nighttime, when the sunlight was missing in it. We were continuously exposed to that form of infrared in some capacity to maintain what I call our water battery. The mitochondria, our battery, we also have this water battery as well.
Yeah, well, look, I think there is definitely the case that we do not fully understand the role of water in this situation. If you look at in atmospheric light, water absorption, it occurs at certain places.
Right.
Like occurs at certain places. Now, I think that those are places where red light is not going to work terribly well. Right? But I am not going to be the person that goes nanometer by nanometer by nanometer testing that. That's brainless. But I think what it does mean, it means that if we want to deliver light in a therapeutic sense, maybe we shouldn't be giving specific wavelengths of light. Maybe we should be giving a continuum in the infrared. Now, Scott Zimmerman's quite important here because, he's got his unique little light bulb. His light bulb, which is an LED light, but in it, he's got a little tungsten coil. And the tungsten coil, because it runs at low voltage, it doesn't really burn out and it doesn't use vast amounts of energy. So I talked to Scott weekly, and actually I said to him end of last week, I think, I said, send me 50 or send me a chunk of them and we'll run tests on them. Now, I'm doing that because my thoughts have evolved. I realize now that it is not about a specific wavelength range. We've got to recreate a broad spectrum, and that broad spectrum is a sunlight spectrum. And Scott, I think, is the closest person to doing it. And also, he's got a commercial interest, but he's also got a fundamental academic interest in what's going on. It makes him very easy to work with. So, yeah.
Yeah, no, absolutely. I agree. We can't cherry pick wavelengths and just say these are the most important ones. That we evolved under the full spectrum for a reason, and perhaps we don't know the reason behind each wavelength, but there certainly is the potential for it to be biologically impactful. And for us to think we know better, I think is a little egotistical, right? So I love where you're going with the idea of full spectrum light. So then with those particular bulbs, are you planning on doing very similar blood glucose research on them, exposing an office environment essentially to that type of light that has the near infrared as well, the red and the near infrared, and seeing what types of impacts it has?
My first thing I'm going to do, which is simple and it's easy for me to do and easy for me to get subjects, is to wheel people in, give them a 10-minute test on their retinal function, then give them a light bulb. Now, we don't do this in lab environments anymore. We've got to move to a more natural environment. So I give them the light bulb and I say, go home. Use this light bulb generally in your environment. Try to have it on during the day behind your computer. Come back a week later and we'll test you. My testing is completely non-invasive. So I can get subjects for that. So that's my first target. We get a positive on that target, Yeah, blood sugars, definitely. And we've got to start looking for other metrics. So we know if we give red light to flies, they move better, right? There's some strong indications of improved cognitive ability with them. We've got to start translating that sort of thing. So I've got a pile of mats outside my office, and we have been running experiments getting people to stand on one foot with their eyes closed. That's a really good one, because you just said, how many seconds can you stand on one foot with your eyes closed? So can we do that by changing the lighting? So vision's important. None of us like to lose our vision, but also mobility in older people. You break your hip, there's a big problem. A really big problem. So balance and mobility is certainly something we're shifting over to, but not in tough experiments, but in experiments where we change people's light in their living and working environment. I think we've come to the end of saying, I'm going to shine this light in your eye for, 15 seconds. We've gone beyond that now. We're in a much more translational stage.
Which I love. And I think, I think that gives the practical application is, hey, get a bulb like this in your environment, right? Which is a pretty easy thing for most people to do. So no, and I love that as well, because the ways to test. So then is it safe to say that you're assessing mitochondrial efficiency in different ways? So through the eyes in terms of visual acuity, through the movement based on proprioception and muscular strength and balance. Could you do something like grip strength as a means of seeing? I mean, are there there's other markers of longevity we know that we could, but the mitochondria are the underlying thing that we're thinking is being impacted by this light exposure.
Yeah. Now, there are a range of things you can do. My guiding principle is, what is going to be the easiest way to get you to walk into my office, right? So pricking your finger is not good, right? So standing on one foot is good, because I can say, can you come in here? I just need you for 43 seconds. I look at you and I think, yeah, you're at this age. We've got some rough idea how long you can stand on. So we have to do simple things to get numbers, hard numbers. I'm not asking people to fill in a form saying how well they feel. I'm not interested in that. I'm interested in us saying, you can now stand on one foot 15% longer than you could before. And from that, I'm going to make assumptions. I'm going to make assumptions that there are mitochondrial changes across your body, and that on the whole, they're good, and they fit into that big thing, which is at the end, which is health span, right? They're just little Lego bricks in that health span scenario.
Glenn, what you're doing is brilliant, because I think when people see improvements like that in the before and afters, they themselves are now wanting to continue with the quote unquote therapeutic strategy of light in their environment.
Yeah, so definitely.
It's very brilliant, very translatable. Sarah, anything that you want to chime in here throughout all this conversation? Or are you just soaking it all in, like I was doing with Glenn's stuff?
I'm like, but you keep grabbing it, and I'm like, it's perfect. Yeah. I'm just loving it. And I feel like there's so many implications for people, and just understanding why, I think intuitively when people are in these really blue lit fluorescent light environments, they just don't feel well. They feel more tired, they're more drained, they're more exhausted. And this work, I think, is just so brilliant, because now we actually have empirical data to show what that sort of environment is actually doing to the mitochondria, which is impacting the energy management system of the entire body.
And I would love, I'm going to do that this next summer, is you take someone sitting in a miserable office. And this office looks pretty miserable. I've turned the lights on so you can see me. Normally I've got very low level lighting.
Same.
And actually just take a very small blood sample from those people. Then get them to go out. I've got a small park across the road. Sit in that small park for 20 minutes, because you suddenly start feeling good, right? Even though it's a crappy park in the middle of an urban area, you start to feel good. Take another blood sample. What is it about feeling good in the body? Can we pin that down? Now, that's an experiment I don't like doing, because it's a fishing expedition. But I cannot help but believe that there's going to be something screaming at us when we compare those two blood samples. right? And again, that's taking it away from the lab, taking it to the world that we live in and operate in normally, which is, key target.
No, sure. I can't wait to see those results. I'm super excited for that test. So really quickly, this is always kind of piqued my curiosity. I know why the blue wavelength band of light that you used in a lot of your experiments was 420 nanometers. Is that correct?
420 to 450,
yeah. Which, as you indicated, it's the band that mitochondria really can absorb and interact with, and it is the dominant band that you see in LED light bulbs these days.
Yeah, definitely.
Now, what made you pick the 760 nanometer for red?
Okay, the 760 nanometers, I picked.
I'm sorry, 670. So I don't want to misspeak. 670.
Okay. I picked up the 670 because everybody else was doing it. Right. So the point was, I told this story a number of times. I reviewed a paper about brain damage in animals and how it was improved by 670 nanometers. I was completely confused. I had no idea. I went around asking people, what's this 670? Is that a Christmas light? Is it a bike light? And then I realized that there'd been this bit of science chugging away that I'd been unaware of. Chugging away in Russia, chugging away in Australia. And so we pick that up, and you get caught in a bit of a rut. Now, I know that's a rut now. We've exhausted, I think, a lot of our 670 experimentation. So when we're giving light now, I'm not giving 670 because 670 floods your world with red light. You don't want to sit somewhere with reading a book and the world's flooded with it. So what we're now using is 850 nanometers. You can't see that. And that's one of the wavelengths that goes through your body. Okay, that's one of the ones I pick up on the other side. It is as effective as 670, and it's non-invasive.
Fair.
So some of my lighting, we're now negotiating about lighting in clinical environments, critical care units. If we've got a critical care unit, for whatever reason you're in that critical care unit, whether it's primarily because of a mitochondrial problem or secondarily because mitochondria are effective, I think it's a winner to actually change the lighting. So having longer wave lighting, 850, is the obvious thing to do. We know it's effective. So one of the experiments that is a manuscript sitting almost ready to go, takes me forever to finish these off, is we've exposed people to 850 nanometers and we just wrapped their head in silver foil. They couldn't see a thing, right? A little hole to breathe through, right? So that, and their vision improved very significantly. So the point is that your body is responding to light, right? It's responding to the light. My improvements in vision were partly because the retina got exposed to long wavelength light, but the body can do it on its own. And so I think with that in mind, we're going to move over to wavelengths of light where you walk into the room. So we've got a big 850 panel, walk into the room, give people a cup of coffee, have a chat for 10 minutes, test their vision afterwards. It improves. Okay, wow. It improves. So we're giving you back that longer wavelength light. We're moving further out to a longer wavelength. Sometimes it's also determined by what LEDs you can get that are decent. Some LEDs are just absolutely awful. 850, there's a lot of 850 LEDs around. They're relatively good. And a lot of our devices we're building now or putting together are focused around that and thinking about the clinical environment for them.
Brilliant, brilliant. Yeah, I love this. So what I'm hearing, let me make sure I might translate this for people. What I'm hearing is that it's not necessarily site-specific exposure to 850 nanometers that derives a benefit. You can essentially cover someone's eyes, yet have 850 nanometers in their ambient environment and still show improvements on the various parameters that you're testing.
Definitely. And people have shown that in animals. You know, the Australian guys who were very much the lead in this field, they shone red light into a mouse eye and they got improvements in it. And then their control was to shine it on the back. And they got really disturbed because they got 50%. If they got 100% improvement in the eye, they got 50% improvement on the back. This was disturbing, right? This really, we were fighting off the accusations of polishing crystals, you know, and it all being very, very woolly. And that I did say to people, can we keep quiet about this for a while till we know about it? But we do know that when we shine a light on different parts of the body, we change cytokines in the blood. Everybody goes, oh, cytokines, they mustn't go up, they're awful. But in actual fact, small changes in cytokines are very important signaling molecules. So this has given rise to the fact that if I want to treat a part of your body, I do not necessarily have to shine the light directly on that bit, okay? So when you're on the beach and you put a towel over your face, you're still getting a therapeutic dose. It doesn't have to be through your eyes.
But we actually get that question. So Sarah and I have private communities all the time. It's like, am I ruining everything with my eyes are closed? It's like, no, you're not. Your body is way too intelligent.
Yeah. And also the other thing is that the published stories are actually very inaccurate. Long wavelength light goes through your lid very effectively. And the reason we wrapped people's heads in silver foil was because when we turned the long wavelength light on, the 850, when this wall panel, we went around measuring it and it was bouncing all over the place. You know, it was, so I thought, well, let's turn their backs to the source and we'll have them facing us when we had our cup of coffee, you know, with their backs to the, and then we got really panicky because I went around and you know, it's being reflected off black walls. There's very little in the world. There's very few pigments that absorb at things like 850. So it's got nowhere to go, nowhere to hide. And it bounces around, just bounces around. So you know, the only way out of it was silver foil.
Well, it bounces around until it finds a human body, right? Or you know,
I could have said that, but you know, I'm happy that you've said it. Yeah, it finds an absorber.
Sure.